we've had a program at Voyager for several years looking at the targeting tau as a treatment for Alzheimer's disease. We started with pathological Alzheimer's disease material and immunized animals to identify antibodies that would target pathological forms of tau. We've screened several hundred candidates, and through that screening, we identified 4 novel antibodies that met particular criteria for advancement. Those criteria involve selectivity, affinity, functional inhibition of pathological forms of tau in vitro and in vivo, as well as developability. Three of those antibodies targeted the same epitope, and that's in the C-terminal region of tau. That's actually pretty important. There are a variety of tau antibodies that have been tested in the clinic, or some are currently in evaluation. And those that have been tested previously and have not succeeded have targeted other regions of tau. So we feel that the C-terminal targeting is pretty important. The other aspect that we find very important is that the data that we see from our preclinical animal model is remarkably robust. So the inhibition of tau pathology in our animal models that is evoked by the antibodies is quite strong. This model is a bit of a complicated model. We try to stay as close to the clinical relevance as we can. So we actually inject a little bit of pathological tau into the brains of a mouse, and then we look at the ability of our tau antibodies to prevent the spread of that pathological material. And our novel antibodies do that quite well. And so we're very excited by it. We're excited by the ability that we've had to identify our lead humanized candidate. We've identified a lead candidate, and we're moving forward with that candidate, targeting an IND in the first half of 2024. We've seen, again, it's in this particular set of animal models where we are looking at the pathology, the spread of pathology of tau. Now, this is an hypothesis that has been growing over the years, where if you look at pathology in patients with Alzheimer's disease, this tau pathology spreads to the brain and is pretty closely correlated with the progression of the disease and with the cognitive decline. And so that gives us an opportunity in clinical trials to use PET tracers as part of that readout. Our animal models, we look at the exact same thing, not necessarily with PET tracers, because we can look specifically in the brains of the animals. But what we do is we test our antibodies to see for their ability to block that spread. Our lead antibody, the preclinical tests that we've done on the lead antibody indicate upwards around 70% of inhibition of the spread of that pathological material, which is quite robust and something that we're very excited about.